Bismillahirrahmanirrahim. I am Rehman, lecturer, Department of Zoology. Dear student, this is the first online recorded lecture of the subject of zoology, and today we discuss about metamorphosis in amphibians. Our discussing contents include meaning of metamorphosis, discussing its various types, what sort of metamorphic changes occur in amphibian during metamorphosis, what are the morphological metamorphic changes, biochemical changes occur during metamorphosis, what sort of changes occur in physiology, and hormonal control for metamorphosis. Lecture outcomes. After watching and listening this lecture, dear student, you will be able to know the meaning of metamorphosis, types of amphibian metamorphosis, structure and hormonal control. What is the meaning of metamorphosis? Meta is a Greek word which means changing its nature, place or order, and morph means form or structure. Metamorphosis may be defined as a rapid differentiation of adult character after a relatively prolonged period of slow or arrested differentiation in a larva. According to Rehman and Trupp, metamorphosis can be defined as a radical transformation from larval life to the adult stage involving structural, physiological, biochemical and behavioral changes. Dear student, the life history of some invertebrate including insects and echinoderm and vertebrate including amphibian and fishes are characterized by the process of metamorphosis. It is the process of abrupt changes in form or structure during the life cycle of amphibians. As in the given diagram, you can see that a frog life begins as a fertilized egg. A female frog lays a lot of eggs at one time in a pond. These eggs soon hatch into a tadpole larva. When tadpole hatches, it looks more like fish than a frog. It does not have any legs. It has gills that allow to breathe. The tadpole grows two front legs as you see in the diagram, and its tail becomes shorter and shorter. In adult stage, tail will eventually disappear completely and it will start to eat insect. Young frog will grow for about 2 to 4 years to become an adult. Types of amphibian metamorphosis. There are two types of amphibian metamorphosis: progressive and retrogressive metamorphosis. In progressive metamorphosis, animal progresses in evolutionary grades. Progressive means favoring or promoting progress or gradually advancing or progress towards improved condition. This metamorphosis is considered as progressive metamorphosis. These types of metamorphosis mostly observed in the order L09s of amphibians. Retrogressive metamorphosis. When metamorphosis takes place in lower direction, when an animal retrogresses or shows indication of degeneration in the scale of evolution called retrogressive metamorphosis. This is an attribute of Urochordata where an aquatic free-swimming larva with notochord change into sessile non-chordate adult. Metamorphic changes of amphibians. Amphibians divided into 3 stages: pre-metamorphic stage, pro-metamorphosis and metamorphic climax. So first of all we discuss pre-metamorphic stage. This stage is characterized by the considerable growth and development of larval structures, but metamorphosis does not occur. Usually this stage lasts for about 7 weeks in a common typical frog. It is the growth period without much changes in the body. However, in this phase, emergence of hind limbs start. Second, pro-metamorphosis. This stage is characterized by the continuous growth, especially the development of limbs, fore and hind limbs, repositions of eye, thinning of operculums, muscular jaws develop. In this phase, tails and gills are completely reabsorbed. Third, metamorphic climax. This stage is characterized by the radical changes in the feature of larva, and climax is considered by the loss of most larval feature. The larval feature that are present including gills, nature, herbivores, nictitating membranes and excretory material, ammonia and thin skin, these are totally replaced. Gills are replaced by lungs, herbivores change into carnivorous, nictitating membrane that are absent in the larval stage, present in the adult stage during metamorphic climax, and ammonia change into urea, and thin skin change into thick, and mucus gland appear during metamorphic climax. Structure of a metamorphosis. Structure of freshly hatched tadpole larva. A freshly hatched tadpole larva has a limbless body. The body is divided into three parts: an ovoid head, a short trunk, and a slender tail. A small opening situated ventrally at the root of tail is known as anus. An adhesive sucker is present on the ventral side of the head by which the tadpole larva attach itself to the aquatic weeds. The mouth is lacking, and as a result it cannot take anything from outside. The yolk material provides nutrition. The respiratory organ comprises of three pairs of highly vascular and branched feathery external gills. After a few days, the mouth is formed near the sucker. A pair of horny jaws surround the mouth. The tail becomes more elongated and develops a dorsal and ventral fin. V-shaped myotomes develops on both sides of the tail. Tadpole larva ingest aquatic weeds, as a result of which the alimentary canal becomes extremely elongated. To accommodate such a long alimentary canal, inside the cavity of the short trunk, it becomes spirally coiled. Structure of an advanced tadpole larva. In the advanced stage, pharynx of the tadpole larva become perforated by gill slits. External gills disappear and the internal gills are formed between the gill slits. The gills and gill slits are covered by a membrane known as operculum, or it is also called gill cover. Thus the tadpole larva has three pairs of external gills at the start, which are subsequently replaced by three pairs of internal gills. In the larval stage, the arterial arches also show modification in terms of both external and internal gills. The operculum fuses with the trunk on all sides except a small opening called spiracle on left side. Water enters into the pharynx through the mouth and goes out through the spiracle. During this transit of water, the internal gills are bathed with water containing oxygen dissolved in it. While the internal gills are functioning, a pair of lungs develop as outgrowth from the pharynx on ventral surface. The hind limbs appear prior to the forelimbs. The forelimbs remain first hidden under the operculum and subsequently emerge through it. Structure of a freshly formed toad. After the absorption of tail, the young toad leaves the primal aquatic home and comes to the land. The mouth becomes wider and a pair of true bony jaws replace the horny jaws. Change its food habit to become carnivorous type. As a result, the alimentary canal becomes short and less coiled. The change that takes place in the tadpole can be divided into four groups. These include changes of tadpole in habit and habitat, morphological metamorphic changes, biochemical changes during metamorphosis, and changes in physiology. Changes of tadpole in habit and habitat. With the metamorphosis, the metamorphosed larva leaves aquatic mediums and frequently visits the land. The herbivorous tadpole larva change into carnivorous, especially consume the insect and it becomes insectivorous. The preying habits developed by the adults, and adult animals become more active and swift moving. In the first stage of adulthood, They jumps into the nearby pond and another aquatic medium and then jumps on land by their elongated hind limbs. Morphological metamorphic changes. These also divided into two phases: regressive changes and progressive changes. Regressive changes. The tissue of the tail and tail fin are completely absorbed. The horny jaws with teeth are shed. External gills disappear. Length of alimentary canal much reduces. The change of blood vascular system takes place, and ultimately some blood vessels are reduced. The lateral line sense organ, operculum and spiracle disappear. What are progressive changes? In this case, the fore and hind limbs increase in size. Tongues become long and more elastic. Eyes become large and prominent and develop eyelid and nictitating membrane. External nostril communicate with buccal cavity through internal nostril. Tympanum and middle ear develop. Liver become more enlarged. Three-chamber heart develops from two-chamber heart. Pronephros is replaced by mesonephron. Mesonephric act as an adult kidney. Biochemical changes during metamorphosis. The concentration of serum protein become double during metamorphosis. The biosynthesis and concentration of hemoglobin are greater in adult than in larva. In the liver, DNA synthesis, lipid synthesis, enzyme for urea cycle or ornithine cycle increase during adult stage. Alkaline phosphatase and hydrolase decrease in adult stage of neurons. What are the changes occur in physiology? At the beginning of metamorphosis, the pancreas starts to secrete insulin and glucagon. This is related to increased role of liver during metamorphosis. During the larval stage, the end product of nitrogen metabolism is ammonia. But after metamorphosis, the toad and frog excrete most of their nitrogen in the form of urea. This is shift from ammonotalism to ureatilism with the change of environment from aquatic medium to lands. Ammonotalism is a phenomenon found in those organisms which secrete ammonia as an excretory material, while ureatilism is a phenomenon that is found in those organisms which secrete urea as an excretory material. Hormonal control for metamorphosis. Two hormones such as triiodothyronine and tetraiodothyronine, or it is also called thyroxine, are necessary for biochemical and morphological changes during neurons metamorphosis. These thyroid hormones are produced by the induction of interior pituitary lobes when it reaches a certain degree of differentiation. Then it is capable to synthesize a hormone, thyrotropin, thyroid stimulating hormone, which act on the thyroid gland, stimulate the production and secretion of triiodothyronine and thyroxine. In pre-metamorphic stage, as discussed earlier, the prolactin level is high, but the level of thyroid stimulating hormone and thyroid hormone are low, and the links between hypothalamus and pituitary gland is poorly developed in pre-metamorphic stage. But in pro-metamorphosis, the link between hypothalamus and pituitary gland highly developed. The prolactin level is low, but the level of thyroid stimulating hormone and thyroid hormone are high. In metamorphic climax, the prolactin level increase suddenly, then maintain a steady low level. The thyroid stimulating hormone is high until the end of climax and thyroid hormone level become low. This is the final schematic representation of larva development and metamorphosis in a typical frog. These are the references of the today topic. Thank you.